which identify the genetic factors contributing to complex diseases using integrative approaches with human populations and model organisms. His primary areas of interest have been on cardiovascular disease outcomes, with an emphasis on the major adverse cardiac events such as myocardial infarction, stroke, death, and in primary and secondary care settings. Dr. Allayee attended UCLA for his undergraduate and his graduate MA and Ph.D. studies, followed by 2 NIH-funded fellowships in endocrinology and metabolism, as well as medical genetics at Harvard-UCLA Medical Center, and affiliated with the David Geffen School of Medicine here at UCLA, respectively. He then served on the faculty of USC School of Medicine for over 20 years before rejoining us here at UCLA in 2025. You got it here. Thank you very much.
Okay. I'm assuming you can still hear us on Zoom?
Yes.
Yes.
Okay, great. Thank you very much for the kind introduction. Happy to be here today. So this grand round is going to be slightly different than what you're probably normally used to. It's not going to be as clinical, but I've tried to sort of make it as clinically relevant as possible. And as the title says, can we use genetic strategies to understand major adverse cardiac events, which is obviously very important for you guys in the clinic. So as you heard in the introduction, we like to use multiple levels of data and different types of approaches to understand cardiometabolic diseases. Really where we start mostly is at the beginning is, let me get a laser pointer. So we like to start with human genetic studies primarily, although we can also start in the mouse. We like to identify the genetic risk factors, characterize those variants. That helps us understand the disease process, and hopefully we can take this to either novel therapeutic strategies or use it to sort of manage risk assessment and things like that. You can also do these things starting again, as I said, in the mouse, come into the identify the genes that way, do the same thing, and then take it to humans. What you find in humans, you can take to mouse. And you know, over the last 10 or 15 years, it's not just having genetic variation in clinical outcome, but you can incorporate a variety of other types of data, either at the gene expression level, the transcriptome, at the proteomic level, the lipidome, the metabolome, the microbiome. You can, you know, there's a variety of levels of data. And this is a systems genetics approach that people like Jake Lucis here at UCLA and others have sort of been using for a long time. So that's sort of the framework where we do through which we do our studies. And focusing on heart disease and what do we know about the genetics of heart disease? So this is a genome-wide association study that was published a few years ago. It's, as of today, it's the largest one that has been done. It's had over 1 million participants in it. And as you can see, I'm not sure how familiar people are with this what's called a Manhattan plot, but on the x-axis, what you see is the entire genome starting with chromosome 1 all the way to chromosome 22. On the y-axis is the statistical significance level. So the higher the level, the more significant it is. And each of these dots represents a SNP or a genetic variant that has been tested for association with coronary disease. So here we have about 10 million dots, meaning we tested about 10 million variants, or not we, but the authors of this study. This was a large consortium sort of effort. And what you see here is that there are about 300 regions of the genome that are significantly associated with coronary disease. So you can say that, like, you know, there must be 300 genetic genomic regions that influence risk of coronary disease. That's as of 2025. So what has this really taught us? Well, we know that those common variants that I just showed you on the previous slide, a fraction of them, not the majority, but only about 30% of them are involved in risk factors that everybody knows for coronary disease: lipid metabolism, blood pressure, inflammation, thrombosis, platelet function. But the majority are actually, it's not known how they influence disease risk. And the best example of that is that the first locus that was identified almost 20 years ago, which is on chromosome 9, very strongly associated. That's actually, I can show you right here. That p-value for association with coronary disease is like in excess of 10 to the minus 300. And yet nobody knows how it affects heart disease. It's not lipids, it's not blood pressure, it's not, nobody knows. It's like the most perplexing thing. So, you know, genetics is great because, you know, you go in with an unbiased sort of approach, and so you can pull out novel mechanisms. And there's quite a bit of work to do to understand what the majority of those loci are doing. So that's with common variants. People said, well, we can't really explain all of heart disease by doing these GWAS, so let's just start sequencing people and look for rare variants. So people have also done that, not on the scale of the common variant analyses, but the rare variants, there's some evidence, although not nearly as much a good evidence as there is for the common variants, that rare variants, things that people just carry themselves, and not like sort of circulating in the population, can also influence heart disease risks through the same sort of like, risk factors, blood pressure, lipid metabolism, et cetera. But then also some, are associated and nobody really knows why. This is only part of the picture. Genetics is one thing, but as we know, it's not just your genes, it's also like what you're exposed to. So the environment and things like smoking, for example, is very well known. Air pollution, the microbiome, you can consider that kind of the environment because of like your diet, as well as the interactions between genes and environment. Those can also contribute to risk of coronary disease. And again, through known mechanisms as well as unknown mechanisms. So basically, all of this interplay comes together to take a normal, healthy artery to one that has plaque buildup. And if it progresses to the point where you get rupture, you get a myocardial infarction or stroke, those are the thrombotic events. So despite effective lifestyle changes and pharmacological interventions, you get people that maybe quit smoking, you aggressively lower their cholesterol levels, you manage their blood pressure, et cetera, you still have this, what we call, and that's what you guys know very well, is this residual risk of major adverse cardiac events. This is a paper that was published 8 years ago in the New England Journal of Medicine. I think it was one of the first ones for the PCSK9 inhibitors. And the point here is that even though the PCSK9 inhibitor added to standard of care decreases risk of coronary disease, you still have about 12 or 13% of individuals over 3 years developing MI, stroke, or all-cause mortality. So you can't take this down to zero by just making people quit smoking and lowering their cholesterol and managing blood pressure. There's still something going on. And the question is, can we use genetics to address this residual risk and the risk of these events, MI and stroke? So that's sort of like the background by which I'm going to sort of describe what we've been doing. And obviously, we need to find new drugs, new pathways that helps us understand the biology, but then also can be clinically relevant. So I'm going to tell you about two stories, and I apologize if I'm speaking really fast because I want to get through this, but if you have any questions, please feel free to interrupt me. That includes people who are on Zoom. If you have questions, you need clarification. So the first story I'm going to tell you is a study that we've done with Stan Hazen at the Cleveland Clinic, somebody who we've been working with for many, many years. And that one is that there is niacin-derived metabolites that increase cardiovascular risk through vascular inflammation. The second one I'm going to tell you is not published. Part of it is published, but it's more recent work, and it sort of demonstrates our approach to these large-scale genetic studies. So metabolomics, which is the approach that was used in this first study I'm going to tell you about, usually begins with some sort of sample. Either it's cell culture, it's tissue. This is not so easy to do in humans with respect to tissue, but certainly you can collect bodily fluids like plasma or urine. Even people have done metabolomics with CSF. You take those, you run it through a mass spec machine, you get out the mass to charge ratio spectra, and then you can identify those metabolites, hopefully based on that spectra. Some of them you may not be able to identify, but then you do some data analysis, and then if you're comparing two groups, you look to see what's upregulated, what's downregulated, what's clinically associated, and so it's sort of a window into metabolism. So if genetics is on the far left, or if DNA is at the beginning or at the left, and then clinical outcome is in the middle, metabolomics you can say is somewhere in the middle. It's a slice, a window into the biology that starts from DNA and goes all the way to disease outcome. So Stan has done this sort of metabolomics using plasma from individuals who've experienced MACE over 3 years of follow-up versus those who haven't. This is an angiography-based cohort at the Cleveland Clinic. And has identified many metabolites that he's worked up through functional studies. The best known of these is probably trimethylamine N oxide. Maybe you guys have heard of this. It's derived from choline and gut microbiome mediated metabolism of dietary choline and carnitine. It is associated with increased risk. It affects it through multiple different sort of pathways, maybe reverse cholesterol transport, inflammation, vascular wall effects, et cetera. Another more recent one was phenylacetylglutamine, another metabolite that's derived from dietary phenylalanine, not entirely from the gut microbiome, also has a hepatic-mediated mechanism, and that's also associated with thrombotic risk. Interestingly enough, maybe signals through the beta adrenergic receptors on platelets. So that's kind of an interesting finding. But Stan has systematically been doing this, and we, of course, bring in the genetics and try to understand their metabolism and their biology using genetics. So it's a very nice sort of like collaborative setup. In addition to the known metabolites, what you also find in the metabolomic screens are unknown metabolites that are strongly associated with increased risk of MACE. For example, this one that's highlighted on the first row, this unknown metabolite 221, which has just a mass to charge ratio of 153, strongly associated with increased risk of MACE, but nobody knows what it is. So that now comes the hard part. Once you identify this compound, well, what is it? How is it working? And what's the story? And so that's where Stan's chemistry background and sort of like his expertise comes in. If you just look at the association of, well, what is the basis for that association that I just showed you? So if you take individuals, this is a discovery cohort of about 1,100 individuals who went through the angiography clinics. If you bin people into the highest quartile versus the third, second, and first quartile for this unknown metabolite, you can just look in the Kaplan-Meier curve that individuals who have the highest levels of this metabolite are the ones that are at most risk. That's shown by the Kaplan-Meier curve, and then the hazard ratio is shown on the right. So I'm going to summarize 5 years of work into 3 bullet points. And so it turns out that this metabolite, which has got this mass to charge ratio, is not one, but 2 compounds that has the same chemical composition. And so on the mass spectra, they superimpose on each other. But through a whole lot of chemistry that Stan, did, turns out that these 2 metabolites are these things called N-methyl-2-pyridone 5-carboxyamide and N1-methyl-4-pyridone 3-carboxyamide. 2-py, 4-py. I'm not going to even talk about the other ones anymore. 2-py and 4-py. And not much is known about these 2 metabolites except that they are produced from niacin. As you guys know, niacin, is vitamin B. It's used to raise HDL, et cetera. So I'm going to come to that in a second. But this signal that you're seeing, this clinical association, is due to these 2 metabolites. So Stan figured out an assay to actually distinguish between these 2. And in a validation cohort, which is 2,000 subjects independent of the first, he was able to quantify 4-py and 2-py separately, and they both show this association with increased risk of MACE. So it validates the clinical association, but actually focuses on each metabolite. And again, you can see the Kaplan-Meier curves up at the top with the hazard ratios down at the bottom. So whether you have 2-py or 4-py in the 4th quartile, you have about a 2-fold, 1.5 to 2-fold increased risk of having MI, stroke, or death over 3 years of follow-up. So that's the clinical epidemiology. The question is, can we understand the biology? And so that's where we came in, and we did a genome-wide association study, just like I showed you for that published genetic analysis for coronary disease, but we did it for these 2 metabolites. What's shown here is not a Manhattan plot. We call this a Miami plot, because what you have is the GWAS results for 2-py going sort of like up the page, and then you have that superimposed with the GWAS results for 4-py going down the page. And what kind of is striking is that there's really no loci in the genome that are associated with either metabolite except this one locus on chromosome 2. And you don't see the resolution here, but it looks like it's exactly the same locus that's going in opposite, that's being associated with both metabolites. And in fact, it is, because the SNP that's up at the top that is associated with 2-py gives a p-value of 10 to the minus 41, and it's that same exact SNP that's going and is being associated with 4-py and gives a p-value of 10 to the minus 12. We assume that the difference in the p-values is simply because there was 37,000 people in the 2-py analysis, but only 11,000 in the 4-py. So it's just a sample size issue. They're probably equivalently associated if you had the same number of individuals. So this SNP that's on chromosome 2, we've labeled the nearest gene as ACMSD. There's multiple genes in there, but this SNP is like pretty close to ACMSD. So that was our sort of first candidate. What is this gene ACMSD? What is this genetic variant doing to ACMSD? And how does that translate to an association with these metabolites? So if you use publicly available gene expression data and you go and you ask, what are the 3 genotypes for that SNP that we found? There is TT, there's TG, and GG. Well, individuals who are TT have lower gene expression of this ACMSD gene in the liver. So the T allele reduces ACMSD expression, but is associated with elevated 4PY and 2PY levels. That's shown right here. So the lower you have ACMSD, the higher the levels of these metabolites. What does that mean? Well, the first thing was to see, like, well, is this even true? Does this model make sense? Is it ACMSD that's the gene? So you can go into the mouse and you can manipulate ACMSD expression both at the mRNA level and at the protein level. You can knock it down, in this case by about 50%. And when you knock it down, what you see is an elevation of 2PY and 4PY in the circulation. So this model seems to work. In humans, you have lower ACMSD, you have higher 4PY, 2PY. And in mice, if you perturb ACMSD and lower its expression, you also get higher 2PY and 4PY. So it's consistent. Decreasing this enzyme increases the metabolite. Why? That's the next question. Why? So this is a very complicated biochemistry sort of pathway, but I'm going to kind of keep it simple. It turns out that ACMSD, which is just shown right here in the middle, is involved in the metabolism of tryptophan. So tryptophan, dietary essential amino acid, can go down multiple pathways. One of the pathways that it goes down is the kynurenine pathway. It ends up going to kynurenine right here. And then from here, kynurenine can go either this side, and we're going to ignore that branch, but what it can also do is it can come down here and through a series of reactions ends up on this metabolite, and I'm just going to call it ACMS, this amino carboxymuconic acid semialdehyde. ACMS then is metabolized by this ACMSD enzyme to this branch down here, and this goes and feeds the energy metabolism, the tricarboxylic acid cycle. So this is the branch through which ACMSD functions. Okay, it takes this ACMS metabolite and it pushes it down this way. So if you have reduced activity of ACMS, then you would maybe push ACMS through a non-enzymatic pathway towards quinolinic acid and NAD synthesis, which is the other branch. So this is a non-enzymatic reaction. This branch right here is the enzymatic reaction. So now we're saying that, well, there's less ACMSD and increased metabolites. How is that related to this? So let me simplify this figure for you. So again, here we have tryptophan. There's that intermediate metabolite ACMS. There's ACMSD that will push it down towards the TCA cycle. And if you presumably decrease ACMSD genetically or experimentally in humans and mice, respectively, you could push and shunt the increased production of quinolinic acid, which would then feed to NAD. And it turns out that 4PY and 2PY are the very downstream products of NAD catabolism. So the more NAD you have, the more 4PY and 2PY you will produce. So maybe the reason why we're seeing in humans variants that decrease ACMSD be associated with 4PY or be associated with increased 4PY and 2PY is because we're blocking this sort of pathway down to the TCA cycle and we're shunting everything towards NAD and nicotinamide metabolism. So that would be the biological model that we've come up with.
Which therefore increases 2PY, 4PY.
Which then therefore increases 2PY, 4PY.
Like CD38, you knock that out?
Yeah.
That decreases the production, the degradation of NAD to nicotinamide?
Yeah.
So that blocks out the growth.
But there's more to it. There's more to it. Because that's in mice. And so what happens is that maybe in mice, under sort of controlled dietary conditions, that might work. But what is also part of this is that you have this pathway. What also can happen is that you can have dietary fortification of niacin feed into this pathway. So it's not just increased NAD because of this, but then there's all this niacin that's coming into the diet and you're flooding the system with a lot of nicotinamide and then you're flushing it out through 4PY and 2PY. And in that context, maybe the effect of 2PY and 4PY would be there. So if you
think that wouldn't that minimize the effect of the polymorphism though?
Well, we haven't done a gene diet interaction. Yes, you could do that. But if you feed your mice, if you're CD38 knockout mice, a lot of niacin, or, and you
Yeah.
fortify them, you might actually overcome the protection that you're seeing because now you're going to be flushing down towards 2py and 4py. So this is not just like genetics. There's also this thing with diet that kind of feeds in. And so I'll come back to the end of this, like this diet fortification and is this really bad? Is it not bad? Okay. So here we go. So if this model is correct, then that same genetic variant that we saw was associated with increased 4py and 2py should be associated with increased intermediate metabolites like quinolinic acid, nicotinamide, methylnicotinamide, etc. So to test that hypothesis, we went and used a publicly available metabolomics GWAS dataset. And in fact, that RS104, that SNP that I mentioned to you, is associated significantly with both quinolinic acid and methylnicotinamide. So it kind of is consistent. You have genetic variant, decreases ACMSD, increases flux towards quinolinic acid, methylnicotinamide, the breakdown products at the end, 2py and 4py. Okay, so then here's a model, again, Jake, so this is in a mice, it's kind of consistent with all this. This group right here went and didn't knock out, like there's a delay with my laser, didn't knock out ACMSD, but they made a mouse that had a whole lot of ACMSD. So the mouse has a whole lot of ACMSD, and it is fluxing away from nicotinamide. So let me go back here and sort of explain it. So instead of blocking over here, imagine you have a lot of ACMSD activity, and you're going to flush things towards the TCA cycle. Well, that's going to deplete you of niacin, of nicotinamide. And that's exactly what happens in the mouse, unless you fortify the mouse with vitamin B. If you don't give that mouse vitamin B, they become nicotinamide deficient, NAD deficient. So this is all sort of like fits together, this biological model. And so now the question is, okay, well, we have the biological model that explains the genetics, but what I haven't told you is like, well, how does it associate with increased risk of cardiovascular events? So we went back and we said, well, what else is this SNP associated with at the level of proteins, not at the level of metabolites? And one of the ones that caught our eye was VCAM, so vascular cell adhesion molecules. So that same SNP that's associated with increased 2py and 4py is also associated with increased VCAM levels. And why is that important? And why would that be interesting? So if, for many of you already know that as part of the atherogenic process and the inflammation that occurs in the artery wall, you get increased expression of VCAM on the luminal surface of endothelial cells. This promotes increased monocyte adhesion and infiltration into the subendothelial space. And so VCAM is known to play a central role in the atherogenic process. So the SNP that we find that's associated with 2py and 4py is also associated with VCAM. So now we're layering in another piece of data in the middle between genetics and clinical outcome. SNP increases metabolite, increases VCAM levels, and could that be associated with cardiovascular disease? Or explain.
That was VCAM.
What's that?
I mean, what was it? Human studies or?
Yeah. That was it.
So what was the tissue that they examined?
This is circulating soluble VCAM levels.
Oh, soluble.
Soluble VCAM, yeah. So this is a proteomics data set. This is actually UK Biobank proteomics. Yeah. Okay, so then if this is the case, can we experimentally prove this? And so then Stan went and took human umbilical vein endothelial cells and incubated with either control vehicle, LPS, which is a positive control and known to induce VCAM for sure at both the RNA and the protein level. And then he independently tested 2py and 4py. And what this data shows is that, well, biologically, it looks like 4py is the compound that is really driving this because 2py doesn't seem to increase VCAM at the RNA level or the sorry, there's like a delay here, or the protein level. But 4py significantly increases VCAM on the endothelial cells. So now it's not both metabolites. This data suggests that maybe it's only 4py that we should be worried about. 2py is coming along for the ride because they're so correlated with each other. The R values, they're both like over 90% correlated. So 2py appears to be like sort of a benign passenger, but 4py looks, at least in vitro, to be the one that's driving this sort of VCAM regulation. can we validate this further in vivo? And so then this was an experiment that was done with mice where you inject 2-PY or 4-PY, and then you visualize VCAM expression on the endothelial cell surface of the vascular wall. You can use CD31, which is a positive control that stains endothelial cells, and under sort of control exposure conditions where you just inject saline or PBS, what you see is you can visualize the endothelium very nicely using CD31. But VCAM is really, not really expressed. I mean, it's there, but it's not like, lighting up. When you inject 2-PY, again, you have very nice, you can visualize the endothelium, but you don't really see increased VCAM on the endothelium. When you give 4-PY, you actually now see the increased expression of VCAM in the endothelium. So this, again, is consistent with the in vitro specificity of 4-PY. And then if you do a sort of a visualization assay, this is like you inject 2-PY and 4-PY, and you can visualize under a microscope the monocytes binding to the veins of the ear in the mice. This is an auricular venule assay. And so what you could see is you have a vehicle, there's no fluorescence. With 2-PY, you don't really see fluorescence, but with 4-PY, you see these monocytes adhering to the endothelium of the auricular veins. So this is in vivo validation further that it's not just expressing VCAM, but it has a functional effect because you're getting more monocytes adhering. So this is all sort of nice, and maybe this is like the explanation for why what started with an untargeted metabolomics finding ends up coming down to sort of potentially a biological mechanism. Now, what does this mean in the end? Well, okay, so here's what we have. We have dietary tryptophan that goes in, and this is like sort of Stan's figure where he's got like a, sort of like a plumbing picture. But you've got this bucket of tryptophan coming in from the diet, and you can go through the sort of ACMS. If you block the ACMSD valve through genetics, you're going to take ACMS and dump it into the nicotinamide pool. Then what you're going to get is dietary influx of the nicotinamide precursors, niacin and things like that. And so now your nicotinamide pool is full of all this stuff. And then that could flush more of the 2-PY and 4-PY out. And then that could be associated with increased risk of MI, stroke, and death. We always sort of like Stan's graphics because he here put a tombstone with like an RIP on it to depict death. It's kind of funny. Anyway, so the question is this: given that dietary niacin is in excess, could increased 4-PY be part of the explanation for why large clinical trials with niacin have failed to reduce risk of CVD? You can raise HDL levels effectively with niacin, but as far as I know, the largest clinical trials that came out in the last 5-6 years really didn't show any benefit. And could this be part of the explanation? Yes, you're giving niacin, you're increasing HDL, but at the same time, you're also increasing these downstream metabolites. And so Stan was trying to get his hands on plasma from the niacin trials to see if, in fact, 4-PY, 2-PY did really spike, and that maybe could be the explanation. Now, in terms of like sort of nutritional guidelines, et cetera, how did this fortification start? So, before in the early 40s, people were vitamin B deficient. And so you had like, a death from pellagra. And so the government mandated fortification with niacin. And so they started putting niacin in diet. As expected, pellagra deaths plummeted. And now they're like virtually zero. And that fortification guideline stayed relatively constant. And then in the early 70s, they actually increased the amount to fortify. So now, here in 2010 or 2025, individuals in the US are probably getting niacin at the upper intake of tolerability. And so given the genetic variation that exists in the human population, are we sort of increasing overall cardiovascular risk because of, like, let's say, this dietary fortification? That's just something to think about. Some people don't want to talk about it. Some people don't like to hear that. Obviously, people who are in the cereal company, because I think they put niacin in cereal and breads and things like that. But this is sort of just a food for thought. No pun intended. Okay. All right. So let me, in the last 20 minutes or so, tell you about something else that we've done. So in this first story that I told you about, we approached the problem of addressing risk of MI and stroke at the metabolomics level. Can we also just do it at the clinical endpoint level? Can we just do a large-scale genetic analysis for heart attack and find genes that way, as opposed to starting with the metabolomics? The answer is yes. And so that's what the second one is. But we've kind of done it in a little bit with a nuance to it. And the reason is because if you think about coronary disease, it starts off with a normal healthy vessel, you get plaque buildup, what we call plaque initiation and progression, and then ultimately is when you get that plaque rupture. When people have done genetic studies, case control studies for heart disease, like the one that I showed you on the first one of the first few slides, they've taken people who have coronary disease but no history of MI, and they've grouped it together with people who have had an MI, and they've called those two groups of individuals as cases. So some people can have coronary disease for their entire life and never have an event. Some people may have an event very early on. But they've just grouped these two together, and they've called those cases compared to people who don't have any evidence of disease. And the basis for this GWAS finding is based on like this sort of like combined group. But it is possible that there are risk factors that are associated with both plaque progression and then subsequently will be associated with MI simply because they contribute to a larger plaque, again, 9p21, some of the lipid-related genes. But it is also conceivable that in the presence of coronary disease, you might have different genetic risk factors that actually push you into having an MI. So they don't have to be the same. Maybe you have specific loci or specific genetic risk for having plaque rupture. Is there any evidence of this? About 15 years or so ago, Murdoch, Riley, and Dan Rader at Penn, and this is a study that we were part of, did an analysis where they compared people with heart disease and MI to just people with only heart disease, and found that ABO blood group, the blood group, ABO, is a risk factor for heart attack and stroke that does not contribute to plaque buildup at all. So that was one of the first pieces of data that said that, well, maybe this sort of question I have here in purple, do distinct genetic risk factors for only MI? The answer is yes. Maybe ABO certainly would fit the bill. Question is, well, is there more? Are there more that we can find using sort of large scale approaches? And so what we decided to do first was to do a genetic analysis on a larger scale than what Murdoch did by, instead of comparing to controls with no disease, what if we compared people who only have CAD to people who have both CAD and MI? So it's kind of a different case control comparison. If we do this, can we find loci that are, because now everybody in this analysis has coronary disease. So if there was a variant that would contribute to coronary disease, it shouldn't pop up in this analysis. We're only going to be hopefully isolating loci that are specifically associated with plaque rupture. So this is an analysis that was done about 5 or 6 years ago. And what you could see here is obviously it doesn't look as robust as that big, large analysis when you've got all those cases, but there appear to be a handful of loci here that are specifically associated with heart attack in the presence of coronary disease. This one on chromosome 12, which has a p-value of 10 to the minus 30 or so, I'll just say that it's a very interesting locus. It's only found in Asians, and it happens to be the locus that is involved in alcohol intolerance. Yeah.
Two questions.
Yeah.
Number one, now a new question of Asians, you mean South Asians? You mean all Asians? And then
East Asians.
East Asians. Okay. First question is going to be, are these patients all MI, like acute MI, like NSTEMI and STEMI, or are these just STEMI patients?
We don't have that level of granularity. It's just if they've had a history. It's like these sort of biobank studies.
Granularity.
But part of it is in angiography-based cohorts. For example, in Stan's cohort, the angiography-based cohort, it was not somebody who actually had active MI at the cath lab. It had to be stable. They got referred to the cath lab for some sort of other thing. They were all stable patients. So there was no acute MI in that one sub-cohort. But the other ones, I couldn't tell you whether.
Interesting if that could ever be disaggregated.
Well, so those kinds of questions, I'll get to this in a second. Maybe we can answer now at UCLA. If you want to find loci that are specific for STEMI versus non-STEMI, maybe we can address that.
Yeah, even plaque rupture MI versus a type 2 MI that's just from demand.
Sure. If you have that information and you have the genetics, that's just how you carve up the pie, right? You can make different comparisons. It's not hard to do. What's that?
You can talk after.
Yeah. Okay. So what this also showed us, aside from this very interesting locus on chromosome 12, is that, well, the usual suspect gene, which was ABO that I told you that Murdoch found before, we actually see it as well. And we see it with even increased significance because our sample size is better. Here is 9p21. It actually doesn't achieve genome-wide significance, and that is exactly what we would expect because 9p21 just builds up plaque. It doesn't contribute to its rupture. And then there's this locus way over here on chromosome 1 where this gene is SLC44A3. We focused on this for a variety of reasons. I'm not going to get into it, but we just said, okay, let's just look at this locus. But one of the things, I'm going to skip over this. One of the things that we did, whoops, if I could just show you. So if we zoom in on that region, you could see that this is the lead SNP. And the reason that we picked SLC44A3 is because the SNP is directly upstream of SLC44A3. So it is associated with SL44A3 expression. But, and to this day, we still look at this locus and like, is it really SLC44A3? Because if you look a little bit further down, there's factor 3, which is very well known to be involved in thrombosis, et cetera. So if you have a locus that's associated with the thrombotic event, and then you have a coagulation factor that's next door, why would we focus on SLC44A3? But we tried our hardest to say that it's factor 3, and we couldn't find any evidence that it's factor 3. So we've sort of like been focusing on SLC44A3. Just a couple points about it. Not much is known about SLC44A3. It might be a choline transporter, but nobody really knows. What is kind of interesting is that another member of this gene family, SLC44A2, has been associated with venous thrombosis. So SLC44A3, venous thrombosis, maybe SLC44A3, arterial thrombosis. That's sort of a reasonable hypothesis. But to ultimately validate it, you would have to do something else. And in fact, we have the genetically knocked out mouse model for SLC44A3. And so we can characterize that mouse if we had a good way of testing for MI in mice. And I'm going to come to that in a second. Okay, another thing about SLC44A3, this is just what's called a single cell RNA-seq or a UMAP plot of coronary lesions in humans. And you can see the different cell types that are found in coronary lesions, endothelial cells, you got the inflammatory cells, you certainly got smooth muscle cells and fibroblasts, et cetera. And SLC44A3 appears to be expressed in the subset of smooth muscle cells. It's also expressed a little bit in endothelial cells, but it's pretty much absent in the inflammatory cells. So if we wanted to hypothesize about how this gene could be associated with myocardial infarction, you might say, well, it could be primarily maybe working through smooth muscle cells and or possibly a little bit endothelial cells, but maybe not the inflammatory cells because it's just not expressed in those cells. Okay, so, and this is the data from like the various papers showing that SLC44A2 is in fact functionally validated for venous thrombosis because when they knocked out 44A2, the mouse had decreased thrombosis. So we could model our SLC44A3 studies on these papers that characterize SLC44A2. So we have some sort of biological insight that we could pursue. Okay, I'm going to skip that real fast because I want to get to this. So that was the first foray that we had into looking at MI-specific loci. Now, fast forward, and because of a lot of publicly available data, a lot of biobanks that are available, we've put this sort of analysis on steroids, what we call genetic analysis of MI-specific loci 2.0. In reality, it's probably like 10.0 because it's got multiple iterations. But what we've done now is cobble together multiple cohorts. GeneBank is the one that's at the Cleveland Clinic, by far the smallest with like only about 2,600 individuals. But we are collaborating with people at the Estonian Biobank. The Cardiogram Consortium is just publicly available data. We have access to the UK Biobank, to FinGen, to China Kaduri, Biobank Japan, the All of Us cohort, the Million Veterans Program, and most importantly, and this is what I was mentioning, we can do this at UCLA, we have access to Atlas now. And James and Yana, who are both sitting to my right and left, through brute force and sheer determination, have gotten GWAS to work in Atlas. So this is hot off the press, and so we're very excited. And so now if we go into the Atlas database and you have STEMI versus this, so you can address that question, assuming you have enough sample size and things like that, but it could be done. So in now, this analysis that I'm going to show you has 2.3 million people in it with about 180,000 MI cases and about 2 million controls. When you do that, the entire genome lights up. So this is about 370 or so loci that are associated with MI. in this analysis, of which it's about 90 of them are novel. So from that Manhattan plot that I showed you in the first couple slides that I said like has about 300 loci, that rainbow Manhattan plot, we've increased the discovery of genetic risk factors for MI by about a third to like about almost 400 or so. Now the question becomes, how many of these are actually specific for MI and not just associated with MI, because all people with MI have disease, coronary disease, but how are they specifically associated with plaque rupture or not? And so you can do an analysis where you can sort of statistical gymnastics where you can look at the effect size for only heart disease versus the effect size for myocardial infarction. And of course, there's going to be a correlation, but anything that falls off the correlation down towards MI, meaning the ones that are green or orange, would be specific for MI. That's what this analysis kind of shows. If you use a very stringent threshold for defining this sort of like differential association, you could see that there's maybe, I don't know, I would say all the orange and the green. The green are the new loci, the orange are the older loci, but about maybe a dozen or so, maybe 15 or 20 loci in the genome appear to be only associated with heart attack. So that's very interesting for us biologically, but I think clinically, because if you can understand why somebody's having a heart attack, you can manage their coronary disease, but you can never predict when they're going to have a heart attack. So if you can address that as well, then maybe we can bring that residual cardiovascular risk that I showed you down a little bit further. Okay, so how are we going to validate a candidate gene for MI? For those of you who don't know, in mice, it is very difficult, if not impossible, to get a mouse to have a heart. They just don't get lesions in the coronary arteries. They get lesions in the aorta and some other arteries like the carotid and the brachiocephalic, but you just don't get really any in the coronary artery. But there is a mouse model that was developed not by Gary Owens, whose paper. This is a paper that just came out of his lab about a year ago. Not by Gary Owens, but by Monty Krieger. I think he's on this paper. Yeah, he's right there, Monty Krieger. So Monty Krieger, 20 years ago or so, generated a mouse that the mouse died from heart attack at 8 weeks of age. So it was too severe of a phenotype. Fast forward about 20 years, and Gary Owens has kind of tweaked the model. And so what you can do is you can have wild type mice over here, and you can have mice that have this mutation in SRB1. And in the presence of LDL receptor deficiency, which is how you make mice hyperlipidemic, what you can see is about at 26 weeks of age, you can get about 70% mortality. And that mortality is because of MI, stroke, and plaque rupture. So now this is a genetic model of plaque rupture that one could use to test these genes that we're finding from our MI analysis, such as SLC. But more importantly, you can also use it to test new drugs or existing drugs, because this is a very robust model, and you don't really have to do much. You just have to breed them up, give them the drug, and just wait to see what happens. Now, why are they dying? And this is just sort of like a busy histology slide, but you could see actually in the coronaries now that they have significant plaque in the coronary, both the right coronaries and the left coronaries. That has not been seen before, really. I mean, so this is, in this sense, if you're getting a lesion in the coronary arteries, that's really nice. And in the mice also have evidence of heart attack because, for example, you see fibrosis, troponin is elevated. And if you look at all the mice and you ask, well, where do they have their heart attacks, right? Do you have plaque rupture in the right ventricle? Or sorry, you can have a lesion in the right ventricle and the left ventricle, even at the apex of the heart. And if you do this and you look in the brain, there was also stroke in multiple regions of the brain as well. So it looks to be a really robust model. And one of the nice things about it, like I mentioned, is you can test it for therapeutics. In this paper, what they did is they took a myeloperoxidase inhibitor and they gave it to the mice. Here's the mice that did not get the inhibitor. They just got the Western diet. And of course, you see the same mortality over 25 weeks. But the mice that actually got the inhibitor, there was about a 60% reduction in mortality. And you can see here in this side that you don't see the fibrosis in the mice that got the compound, whereas you do see it robustly in the regular mice. You see decreased fibrotic area, troponin is down, and if you look now at the percentage of mice that got the drug that had MI, 31% compared to 77%, this is the very first sort of slide I showed you where there was also 75% MI there. So you're reducing MI, stroke, and plaque rupture using this compound. So if our genetic analysis reveals novel compounds, we're going to replicate this figure right here, hopefully, with whatever it is. Okay, so That was a whirlwind tour, perhaps. But I'm happy to answer more questions. There's about 5 minutes left. I know everyone's busy. There's probably people waiting. But happy to answer questions, comments from Zoom. Anyone?
Two questions.
Yeah.
One, even though I'm somebody who can ask you a question anyway, so So, what do you think is the niacin cellular metabolism where, is it endothelial cell metabolism? Because this is circulating niacin, right? It's
Yeah. So
niacin.
yeah. Sure.
Or what do you think it is?
Yeah, so I think niacin gets into the, you consume it, it goes to the liver, and that's where it ends up being a 4-PY, 2-PY. What's that?
They do a liver specific.
No, what Stan did is he injected an AAV with an shRNA that knocks it down. That was kind of like liver specific. Yeah. But yeah, you can make a liver specific knockout, and then if you still see the increased VCAM, then it's 4-PY, 2-PY in the circulation affecting the vascular wall. Yeah.
I already asked James because I said you're not going to share what are the MI specific loci with us.
Where he's in the process of yeah. So
what would you hypothesize or kind of loosely say you think are these dozen sites among the different, like you suggested 5 different pathways?
Yeah. So I think some of them are going to be thrombosis and platelet reactivity and things like that. But I think some of them are going to be at the level of the vessel wall. And Gary Owens and others have said that like there's certain smooth muscle cells in coronary lesions that contribute to plaque stability. And so if you lose, I didn't show it to you in that mouse model that he did, but if you lose those, the fibrous cap and like this integrity of the lesion maybe becomes a little unstable. Maybe some of them, like SLC, we think is doing something to smooth muscle cells, not anything that's circulating.
Yes. What's the solution from 4-PY to the VCAM? Is that an metabolite that's metabolized into something else, or is it an inflammatory?
Yeah, great question. So Stan is actively looking for a receptor of some sort. See if like there's something that 4-PY binds to on endothelial cells, and how does that translate in and have like some sort of cascade of gene expression that results in increased VCAM. We did an RNA-seq analysis, but it's not like this thing like just came right out and said, oh, that's the mechanism. So yeah, but yeah, great question. I just wanted to, I always forget this. That's why now I know why people, when they give talks, they acknowledge the people first, because I always like forget to do it. But I want to really acknowledge James, who's sitting over here, and Yana, who we've been working with for a long time, and then Yi, who was a postdoc when I was at USC, who really got this large-scale genetic analysis going. And James, learned, I think, a lot from Yi and Yana, and now has taken over and sort of what you saw, that 2.3 million person GWAS. 2.3. Okay, 2.35 it's all James's work that he's been doing. So yeah, I just wanted to acknowledge him, as well as our collaborators at the Cleveland Clinic, and of course, all these biobanks that are just once you get your hands on them, then it's a lot of fun. You can ask a lot of questions. So yeah.
So do you have any interest in actually defining biological, whether that's like using iPS cells, if it's conserved?
Yeah. There's so much to do. Geneticists usually say, well, that's kind of hard, so I'm just going to increase sample size and do another genetic analysis. but post GWAS follow-up is a big deal. Nobody does it. Already, you could have 5 careers following up on all these loci and figuring out what it is. What we tend to do is find the ones that are sort of dramatic, like for example, an amino acid substitution that you can go and model in a mouse. You can sort of do the ones that are sort of at the gene expression level. We're not genomicists, so we don't really do that. But if we find something that is an amino acid substitution, and then you can sort of get the knockout for it, put in one allele and the other, and sort of do that. That's kind of fun. But some people do that a lot. Yeah.
I have another question.
Yeah.
The second half where you mentioned smooth muscle cells in the actual lesion.
Yeah.
Are they actually true smooth muscle cells, or are they endothelial cells that go in?
No, those are some of those are true smooth muscle cells because they express the classic, for example, smooth muscle cell actin, etc. Those myofibroblasts and the fibroblasts that I showed you, those are the ones that seem to be transitioning. The endothelial cells cluster, completely separately. Was there another question over here?
Yes, Jesus. Nice talk, Hooman. Now my question is about, so you started your first story with a characterization of microbial metabolites, and then discovering these products, and that Stan didn't really know what they were, and then you ended up identifying that in the niacin. So what's really the relationship with this enzyme and these 2 compounds and the microbial metabolism. And isn't it that niacin actually improves like a gut and microbiota health?
Yeah. So I didn't clarify, but the first two metabolites I showed you, TMAO and phenylethylamine, were initially found as unknowns, and then Stan worked them up, identified them as TMAO and PAG, and then showed that they have a gut microbial sort of like mediated metabolism. Okay. The unknown that led to the identification of 2-PY and 4-PY has nothing to do with the microbiome. The microbiome is not involved. It was just another metabolite that came out of this screen, but didn't have a microbiome.
Presumably, niacin improves.
Maybe, I mean, if those clinical trials of niacin collected stool samples, you could
And the other question is that you propose that this approach could lead to identifying some of the portion of the mortality or morbidity,
the residual
that we don't
risk.
understand.
Yeah.
Wouldn't it be that at the end just helps us to understand better what we already know? Let's say, we know that if we control high blood pressure or if we stop smoking, so we improve cardiovascular health.
Mm-hmm.
But we don't know really the whole mechanisms that are involved to get there. So you will be detecting genes, that you will be detecting gene-environment interactions, and you could just be detecting how is that those therapies that we already know take us there, and not necessarily that it will help us to understand what is an unknown.
Yeah, I mean, in the case of 4-PY, 2-PY, that SNP is not associated with lipids or anything. So like that's what I mean.
Okay, so
It's one of those, whenever we find these loci, whether it's for the metabolite or if it's just for MI itself, the first thing we do is like, is it associated with lipids? Is it associated with blood pressure? Can we get the usual suspects? That's the first thing we look at. And then if it's not that, then we like the SLC locus, no effect with lipids at all. None whatsoever. No diabetes, no glucose, and all of the known risk factors have absolutely no association. So what is it doing then? So maybe it is doing something at the level of the signal. Yeah.
I just want to go back to the single cell data and the genetic mutations that you presented. So how did you identify that? Can you actually say that the fibroblasts carry the genetic mutation? Because if you remember Alex Bick's talk from last week, he looked at CHIP and single cell, but it was a little indirect. He overlaid like mitochondrial, testing.
So you're talking about this, right?
Yes.
Yeah. So we found a SNP. The SNP kind of points to this gene SLC44A3, but we know nothing about SLC44A3.
Right.
So one of the first things we ask is like, if you want to rule in SLC44A3 in a MI phenotype, is it even expressed in the tissue that you're looking at? Because
you're
if
looking at tissues.
Yeah. So this is single cell data from coronary lesions, no genetics.
Okay.
Just simply asking, is it expressed there or not? Because if you looked at SLC44A3 expression, and it was like in the hypothalamus or subcortex, I mean, pick like something else, I don't know, something completely non-MI related, I would probably deprioritize it and start looking at factor 3 or another gene in that region. So that's how we've used this. Can you do genetic variant, gene expression at the single cell level? People are trying to do that.
That's right.
Yeah, you just need large sample size. Yeah.
Okay. Following up on that question, so you use your polymorphisms to identify that gene.
Yeah, or we think.
Right. So are you doing like confirmation, like 3C capture to see if you're that
Yeah, I didn't pull the slide, but I actually had the slide in here, but I'm like, I'm not going to put a genomic slide for this slide, and then I should have put it for you were interested in it. But it turns out that in fact the variant shows long range looping to the SLC44A3 promoter, but it doesn't show looping to the factor 3 promoter. So these long-range genomic interactions. So that's another reason, I didn't tell you, but that's another reason why we sort of moved SLC44A3 up and then sort of like brought factor 3 down. Yes, Jeff.
So question for the first story, I think really interesting how you discovered 2-PY, 4-PY, I think fascinating. I'm just curious in terms of like that cool diagram from Stan Hazen about the clogged phosphate, ramifications of just like not being able to use tryptophan in the TCA cycle. I guess maybe a silly question, but like enough redundancy, like other amino acids that can be used. But like, is that thought to potentially lead to a new phenotype like this? This clogged valve specifically down here?
No, because you can probably, I don't think so, because number one, I know that it says like actually that shows that the spigot is open. And this genetic variant in humans doesn't completely shut down ACMSD. It just tweaks it in a little bit, and when you have large sample sizes, things become statistically significant. Biologically, how much is it really blocking the flux towards TCA? Not much. The true way to do that would be to knock out ACMSD in a mouse, where now you've completely shut that pathway off, that portion, and see if there's a consequence of not flexing to the TCA. But my guess is that multiple things go into TCA, and so this is enough for it. It's such a central component to anything.
That SNP for ACMSD too, like we looked at it for like any sort of phenotype outside of like metabolomics or proteomics, like a hard outcome, like let's say MACE, no association, right? So that effect that you have on those metabolites is not even enough to get you that hard outcome of MACE anyway. So yeah, I mean, what the power calculation said, we needed something like 4.5 million individuals to see. So we're working on it.
That's a good point. That's a good point, because now that you have your 2.35 million, you should go and test that SNP. Because before we were like, when this paper was being put together, we were just looking at the 1 million person GWAS or whatever. Like now it's like 2.5 times bigger. So maybe you're approaching something.
Thank you so much.
Oh yeah.